Hello all, and welcome to Professor Chris Thomas's talk on evolution and the spread of antimicrobial disease. It's of course part of the Darwin Shrewsbury Festival 2022, and we'll be learning about how Darwin's principles are still important even today. Now, just a couple of things before we begin. First of all, if you have any questions, please feel free to ask them at the end. There is a Q&A section, which you'll be able to type them into, and I will present them at the end of the presentation, so please feel free to put them in. And secondly, the talk is being recorded and will be available from the website in the future, so feel free to look at that when you get the chance. But without further ado, just a quick intro. We will start hearing from Professor Chris Thomas.
I'm just trying to, there we are. Okay, so thank you very much for inviting me to talk to you this evening. As Jacob said, I'm going to be talking about evolution and spread of antimicrobial resistance genes. And I just want to sort of emphasize at the beginning the sort of relevance to Darwin. So I'm wanting you to think about how does natural variation occur and how do selective islands, so to speak, imagine they might be in the Galapagos, but they could be in the West Midlands. How do those selective islands create the diversity of organisms we observe and see that appearing continuously? So it's something that's happening right in front of our eyes, so to speak. I thought I would just start by a little introduction to me and my evolution. So I did biochemistry as an undergraduate in Oxford, and I was at Queen's College, and I didn't realize at the time how relevant it was that in my first year I was in this the Florey building, because this guy Howard Florey was probably more important even than Fleming in making sure that penicillin came to medical practice. And he was also eventually provost of Queen's College in Oxford. And so I had that connection right from the start, and I hadn't realized. So I did my undergraduate biochemistry degree, and then I got a scholarship that was funded by money from another antibiotic, the cephalosporin studentship. I did my PhD with Keith Dyke, who was at Wadham College. He died about 3 weeks ago, so it's nice to be able to mention him. And I moved to a postgrad college, Linacre College in Oxford, where I did my doctoral research. And what I was fascinated by was antibiotic resistance and plasmids, and those two things come together in this talk. At the end of my second year, I went to a NATO Advanced Study Institute in Athens, and that was my first conference. And I'm not going to bore you with who everyone is on here. This is me looking rather different, an awful lot more hair than I have now. But interestingly, given that we've just been through the pandemic, when you probably all have heard of polymerase chain reaction, PCR, this guy, David Gelfand, was there at the meeting, and he was the person who was particularly important in actually finding the thermophilic DNA polymerase that is used in PCR nowadays. So I found that very inspiring, and I applied to work with the various people, and this guy, Donald Holinsky, said, yes, do come and work with me. And I got a MRC traveling fellowship to go to what felt like being on holiday for 2 years. This is La Jolla, Southern California, the Scripps institution here, and we actually lived 1 block from the beach, and I walked up the hill to University of California, San Diego every day, and worked very much as a intensive research fellow on understanding plasmids. So someone's got a hand up. Do they want to, do I need to respond to that?
Yes, if you have any questions, please pop them in the Q&A and we'll address them at the end. Just on a side note as well, if you're a pupil at Shrewsbury and you want to have your name called out, please say that as well, as you will just come up as Shrewsbury School pupil.
Okay. And then I was invited to apply for a lectureship at the University of Birmingham, and that's where I've been ever since. So here is our campus. It's changed a bit since this picture was taken, but I find it quite interesting, first of all, that Birmingham was the home of the plasmids I was working on in California. Birmingham was where they were isolated for the first time, and those plasmids encode a structure, a protein structure that looks remarkably like old Joe the clock tower, which it uses for bridging the contact with other bacteria and transferring the plasmid. So whenever I look at pictures of the campus, it makes me think of the process by which genes spread between bacteria. So I want to get you thinking, first of all, what are antibiotics? At a very simple level, they are, by definition, selective poisons. They're poisons, but they work against bacteria, and hopefully an awful lot better than they work against humans, plants, or animals. But quite a lot of the antibiotics that we work on in the lab, it says danger, particularly if a female worker in the lab is pregnant. So the whole point of an antibiotic is that it's highly active, but selective for the rapidly multiplying organisms. Penicillin is a very good example. It attacks the bacterial cell wall, and because plant cells and human cells do not have a wall like bacteria, penicillin doesn't affect them. And another key thing is that many antibiotics are naturally produced and generally by bacteria or other fungi. And so you probably have all heard of Fleming, Alexander Fleming, who made the discovery of penicillin by looking at an agar plate on which he'd spread bacteria that were supposed to produce colonies, but he left them on the bench when he went on holiday, and during that time, at room temperature, a fungus grew. At room temperature, they will grow as fast, if not faster, than the bacteria. And so when he came back and looked at the plate, there was a fungal colony, and he could see inhibition of multiplication of the bacteria. And he actually deduced that something was being made. And many fungi produce bioactive compounds that have this sort of effect on other microorganisms, and they're allowing the fungus to compete with the competitors it has for the food sources that it finds. So one of the things about antibiotics is they are highly specific. So they have specific targets, and those might be cell wall biosynthesis, as I've said, for penicillin, membrane function, DNA replication. One of the most prolific groups of antibacterial agents target protein, gene expression and protein biosynthesis. Here's the RNA polymerase making RNA. Here's the ribosome making the protein by translating the messenger RNA. So I want you to think about one particular antibiotic that we've worked on, for many years, and that's an antibiotic called mupirocin or pseudomonic acid. It's used against MRSA, that's methicillin-resistant Staph aureus, and it has this structure, quite complex, but it's still a relatively small molecule, has a molecular weight of about 400. And we've studied how it's made. It's made up of two components, which initially we thought might be made separately and then joined together, but actually it's made in a continuous process. But what I want you to think about is the fact that it has a very specific target, and that is the enzyme isoleucyl tRNA synthetase, which actually puts isoleucine, the amino acid isoleucine, onto the tRNA molecule that the ribosome needs to put isoleucine into the polypeptide chain, where it's encoded in an open reading frame. So this is, we have a crystal structure, it was done in a lab in the States, but we know how mupirocin binds to the active site of the enzyme, and it binds in this groove. We know how it binds. And one of the things is that it interacts with amino acids in that groove. And if you change those amino acids, you might change the activity of the enzyme, but you also can change the way in which the antibiotic binds. So you can change it so that the antibiotic binds less well, and you can get resistance. So thinking about that, if you put a lawn of bacteria, you spread about 100 million bacteria on an agar plate that contains an antibiotic, in this case, for example, mupirocin, you kill most of the bacteria in that population. But in a very large population, there tends to be genetic variation, and you do tend to get a few bacteria able to grow in the presence of that antibiotic and form colonies. And so that illustrates the natural variation that occurs in large populations and on which natural selection acts. So that sort of gives you an idea that resistance can appear. So the first way that I've mentioned is we can get resistance by mutating the gene and the protein that is the target, the specific target for the antibiotic. For example, streptomycin, you get a mutation in the ribosome that gives resistance. Or, as I've said, you can mutate the target for mupirocin, isoleucyl tRNA synthetase, and you can get resistance. But you can also acquire resistance not by mutating the genes that are in your chromosome if you're a bacterium. You can also acquire resistance by gene transfer from other bacteria. And with respect to penicillin, beta-lactamases are very common enzymes. But I want to, again, get you to think about, in some ways, my favorite antibiotic, mupirocin, and get you to think about isoleucyl tRNA synthetases. So, to think about the evolution of a family of genes that have a common function, I want you to think, well, where might resistance come from? Where might resistance genes come from? And I'm going to show you, in a way, there's more than one answer to that. But if we look at the evolution of the isoleucyl tRNA synthetases, and we can do that by aligning, most simply, the protein sequences for the different genes. And so what I want you to look, if you look carefully here, each line is a different, they're all isoleucyl tRNA synthetases, but they are different from different organisms. So these first three are archaea. The next three are bacteria. The next three, you can probably see S. cerevisiae, that's a yeast. A. thaliana, that's a plant. H. sapiens, that's us. Okay, so, and then again, we've got more bacteria down here. But what you can do in these alignments, you can see that obviously they are related to each other. You've got the same sequence. So when we've got the same color all the way down, they're all the same at that position. So it's quite obvious they've come from a common ancestor, but they're not identical to each other. And you can look along and see that there are differences. But let's move on from the very specific differences for a moment, and just think about the fact that you can use computer programs to actually generate a phylogeny from looking at how closely related they are. And this illustrates the phylogeny of all organisms with respect to isoleucine tRNA synthetases. And I hope the letters round the outside are big enough for you to see. But basically, we've got mitochondrial enzymes here. We've got bacterial enzymes there. We've got archaeal enzymes there. We've got, it says eukaryotic-like, but when you look closely, those are mainly bacterial. And then we've got another lot of eukaryotic-like. Those again are found in bacteria. And then we have the actual eukaryotic, so plants, animals, insects are here. Okay. And the fascinating thing is that, so if you look at the inner circle, then you can see that there are phylogenies here. You've got lines of evolution showing closely related organisms and then branching. Okay. So you can see that this group and this group here, these enzymes are very like or closely related to the eukaryote. And we don't really understand why some bacteria have a eukaryotic-like isoleucine tRNA synthetase, but they do. So there must have been some branching a long time ago that actually meant that many bacteria had one sort and others kept an isoleucine tRNA synthetase very much like the eukaryotic one. But when you dig down into this whole phylogeny, one of the fascinating things is that, can you see the black lines? You probably can't read those, but the black ones represent organisms that actually have two isoleucine tRNA synthetases. And so the green arrow here shows Staphylococcus aureus, and the green arrow there shows Staphylococcus aureus. So in Staphylococcus aureus, you find one of these and one of those. So if you look where they are, the ones here are in the chromosome. But that second one is actually found on plasmids. So that's the point, the first point I want to get across to you, that there's this diversity which you can analyze, but actually some organisms seem to have two genes, one from one phylogeny and one from another, part of that phylogeny, and that having two genes is the result of horizontal gene transfer. This organism has acquired a second gene of that sort by movement from another organism. So I want you to think about the fact, if you're looking for an antibiotic, you're looking for something that's going to kill bacteria, but you've then got to test and make sure it's not harmful to whatever organism you want to treat with that antibiotic, whether it's a human or an animal, a cow, a dog, or a plant. So by definition, if we're talking about an antibiotic, there will be a target, but that target in a human, if it exists, will not be sensitive. So mupirocin was chosen because it did not kill human cells. And that means that the isoleucyl tRNA synthetase in human cells is not sensitive, not very sensitive to mupirocin. So if some bugs, some bacteria have a eukaryotic-like isoleucyl tRNA synthetase, then they're not going to be killed by mupirocin either. So if we go back up to here, it turns out that these organisms and those organisms are resistant. So there are lots of bacteria that are actually resistant to mupirocin because they've got that sort of isoleucyl tRNA synthetase. And we can actually look at where the variations are, and you can see this is one of the key places in the active site, and you can see that those eukaryotic-like isoleucyl tRNA synthetases have a much more similar sequence to the actual eukaryotic human, plant, animal isoleucyl tRNA synthetases than the bacterial sensitive bacteria do. And so one of the solutions to getting resistance isn't to mutate your chromosomal enzyme that's sensitive to the antibiotic, but to get a gene from another organism that's already resistant to the antibiotic. So horizontal gene transfer. I want you to hang on to that idea for a moment. I just want to get you to think about a second point. Where else might you get resistance genes? Where else might they transfer from? And so I want you to think about what I told you about antibiotics coming from, and many of them coming from microbes. Mupirocin is produced by an organism called Pseudomonas fluorescens. If we go back up to here, you see the yellow arrows here and there. Pseudomonas fluorescens is another example of an organism that has two isoleucyl tRNA synthetases. So if you think about which organisms by definition must be resistant to antibiotics, they're the ones that actually produce the antibiotic in the first place. So mupirocin is made by a bug, and that mupirocin will diffuse back into the Pseudomonas fluorescens. And if it didn't have a way of resisting that antibiotic, it would stop growing. So antibiotic producers, by definition, have a solution. They already know that you can get resistance to that antibiotic. And so one of the things we've done in my lab over quite a number of years, and we collaborated with Beecham's originally, and then most recently GSK, looking at this pathway, is the pathway that builds mupirocin. And I'm not going to go into detail about this, but it's fascinating if you had time and wanted to go and have a look how mupirocin is made. It's built up from small molecules like acetate, built up chain bit by bit by bit until you end up with the specific molecule that I showed you earlier. And within that gene cluster, there are many functions, and one of those functions is called MUP-M, mupirocin cluster M, gene M, and it's there because it gives resistance to the antibiotic that it makes, and that is an isoleucyl tRNA synthetase that makes the producer resistant. So a second source could be not things that have just happened to have that eukaryotic like isoleucyl tRNA synthetase, but ones which have to have it because they actually make the antibiotic that targets that enzyme. So that's, in a way, getting you to think about how resistance genes, why they exist, and how they can come into being. And then I just wanted to get you to think for a little while about how they spread. So spread of genes between bacteria can occur in multiple ways. The simplest way is that naked DNA can be taken up by bacteria and then undergo recombination or just enter the cell as a plasmid and start multiplying. But the simplest way is transformation by naked DNA. Second way is they can be carried by a virus particle. So a virus infects a cell, and during its multiplication, it picks up a bit of its host DNA. And so when these particles are released, it's carrying not virus DNA, or not just virus DNA, but also host DNA, and it can carry a gene across. But for me, the most fascinating sort of mechanism for gene transfer are plasmids, which I was interested in as a PhD student, and then went to a plasmid lab in California that was involved in the early recombinant DNA technology. And so conjugation allows plasmids to spread from one cell to another by, well, across a sort of protein tube. And those plasmids can also allow chromosomal DNA to be transferred. Okay? So those are the three best understood mechanisms. And so in Staphylococcus aureus, in MRSA, when it becomes resistant to mupirocin, and here this just shows the yellow color here is a lawn of Staphylococcus aureus growing on an agar plate. And this little circle in the middle is a disc of filter paper that's been soaked in the antibiotic mupirocin. If you put it onto methicillin resistant Staph aureus, it will kill it. So that's this, or inhibit its growth. That's this sort of halo around the disc. But if it becomes mupirocin resistant, you get no inhibition of growth. And when you look in those mupirocin resistant MRSA strains, they generally, particularly the ones that are resistant to high levels of mupirocin, carry that second isoleucyl tRNA synthetase gene, which was called MUP-A, and that is on a plasmid. So plasmids are a major carrier of resistance genes because they facilitate the spread from one cell to another. So this is an electron micrograph showing that a plasmid is often, not all plasmids are circles, but many, many of them are. So this shows that it's circular DNA. And so here's a sort of cartoon showing a plasmid. Here's the chromosome and the idea that plasmid can pick up genes from the chromosome. And the simplest way in which genes can do that is by facilitation through transposons. This is not homologous recombination. This is what is called transposition, and it allows genes to move from one location to another, from the chromosome to the plasmid, and then that allows potentially the gene and the plasmid to move easily to other bacteria. So this shows that process, a gene moving onto a plasmid. It becomes a resistance plasmid, if this is an antibiotic resistance gene. Conjugation promotes horizontal gene transfer to bacteria that don't already have the plasmid or that plasmid. And so you can end up with a mixed population. Some bacteria that don't carry any plasmids, some that carry a plasmid that does not have the resistance gene, some that carry the plasmid that does have a resistance gene. And when antibiotic selection is applied, you get weeding out. The bacteria that are resistant survive. The ones that aren't resistant either are killed or at least grow less well than the resistant bacteria. And that is evolution in action. You're weeding out, you're seeing the natural variation in the population, resulting in the fittest surviving. And again, that's sort of Darwinian evolution in action. So I want you to think about plasmids in a way as a gene train, okay? So this is the plasmid, but the engine that drives the plasmid, if it's a conjugated plasmid, it's got to do two things. One is that it allows the plasmid to multiply. That's the replication function. But if it's going to move through a population, it also needs to transfer genes. And so this bridge is a very complex sort of bridge encoded by many genes that allow the mating pairs to form and allow plasmid to transfer. So that, although I've made it a simple block, it's actually much more complicated. And it's the sort of things I've spent most of my 40 years at the University of Birmingham looking at, the fundamental backbone of the plasmid. But as far as antibiotic resistance is concerned, this plasmid can expand dramatically if it picks up something that makes its host have a selective advantage. And so you can imagine it picks up a resistance gene as by the mechanism I was just talking about. And that we'll call antibiotic resistance 1. So that's the first bit of cargo that it picks up. And imagine that that happens in a hospital, possibly in one patient, but you can think of it sort of being like the plasmid has got to one island in the Galapagos where the conditions are of a certain type, which selects for, in this case, plasmids. but in Darwin's case, finches, that have particular beak types. And so it becomes dominant there and then may spread to a new location, a second hospital. So if it picks up resistance to a second antibiotic, you imagine a problem, a series of infections have been treated with antibiotic 1 for a certain length of time. Eventually, almost inevitably, you're going to have a situation in which resistance appears to a second antibiotic, which has been brought in to cope with the resistant infections, the infections that are resistant to the first antibiotic. So antibiotic 1 works for a while, stops working eventually. You start using a second antibiotic, and that works for a while until the second resistance appears. And because the plasmid with resistance to the first antibiotic was very dominant, chances are quite high that the plasmid that picks up the second resistance gene is already going to have the first one. And so it can move from patient to patient, from hospital to hospital, from selection regime to selection regime. And that successive use of different antibiotics over the years results in more and more cargo accumulating on some plasmids. So that's why I sort of like to think of it as a gene train moving along, picking up different cargoes. And when we look at naturally occurring plasmids, some plasmids were isolated back in the 1950s, and then they were sequenced quite a long time later. But what we know now is that the original plasmid, resistance plasmids that were selected often in Japan, where some of the early antibiotics were utilized, have accumulated more and more resistances over the years. They've actually interacted with each other and become hybrids, but they've evolved over many years. And some things have actually helped that process. For example, large-scale use of antibiotics on farms has actually been quite instrumental in some antibiotic resistances appearing, partly because the sort of antibiotics that were used for a while on farms were not very high quality antibiotics. And they carried within them not just dead organisms from which the antibiotics came, but the resistance genes that those producer organisms had to be resistant. And so those fueled the evolution of these resistance plasmids. And you can actually track resistance genes across the world. And so this is one particular cephalosporin resistance gene, CTXM, and you can look at the variations of that gene. There are different sequence types, and you can follow their spread across the world. And you can also follow the origins of these resistance genes. And what seems to correlate with is countries where antibiotics are used very freely, or places where antibiotics are made, and factories belch out effluent into rivers, which may have sewage in them, are a major driver for the evolution of antibiotic resistance and antibiotic resistant plasmids. And one thing that the plasmid that I've worked on for many years, which came actually from the burns unit at the accident hospital here in Birmingham, when Pseudomonas aeruginosa was a problem, and they were using an antibiotic called carbonicillin, which caused resistance throughout the burns unit. They stopped using carbonicillin, the resistance disappeared, but when they tried to start using the antibiotic again, resistance came back very quickly. So the key thing that people learned from that was that actually antibiotic resistance on plasmids never really goes away. It can go sort of dormant, but somewhere in the environment, they're going to be present and come back when selection pressure is reapplied. So just want you to think about, I mean, I don't want to paint a too pessimistic picture. I mean, we know that it's an arms race between bacteria and clinicians to have antibiotics that work. There are strategies that you can use, such as triple or quadruple therapy, so that if resistance appears 1 in 10 million, if you multiply that by 4 antibiotics being used at the same time, you can reduce the possibility of a resistant organism appearing during a treatment process. And that's why it's so important that you follow the instructions, you actually complete a course of antibiotics. But it's also important to reduce antibiotics when they're not actually needed, reduce the use of antibiotics when they're not actually needed. And that means that things that have been ignored in hospitals, like hygiene, when we had antibiotics that solved everything, need to come back and make sure that spread of infections between patients was minimized. And an awful lot of lessons have been learned about that sort of thing. And we'll all be very conscious of things like hygiene as a result of the last 2 years and the pandemic. But there is a need to find new antibiotics and new targets for antibiotics. And so a lot of work is going on that at the moment. But if you think about what I've been telling you, there are also other sorts of solutions that you can imagine. And one of those things might be, could we actually use what we understand about resistance genes to get rid of them? So I told you that a lot of resistance genes are on plasmids. And I actually know a huge amount about plasmids. So plasmid stability in a population depends on replication and a mitotic sort of process. Many plasmids produce little toxins that act as addiction systems. So we know a lot about how they're maintained. And what we've been doing recently is to find ways of actually getting rid of plasmids and the resistance genes that they carry by blocking their replication and neutralizing their addiction systems that mean that the host cannot get rid of the plasmid. So if we've got a bad plasmid, a red plasmid with multiple resistance genes, we put our good plasmid in, we can displace that so that we end up with our good plasmid that doesn't carry resistance. And then we can actually cause the resistance plasmid, the curing plasmid, to self-destruct. And we end up with a bacterium that doesn't carry any plasmid at all. And one of the things we're trying to do at the moment is actually see if we can apply those principles in farm animals, and then eventually humans. So the idea is that we give a probiotic to a pig, for example. It would go in, displace these plasmids from these red bacteria, and you'd end up with a gut that doesn't carry resistance plasmids. And that would mean that it'd be much easier to treat any infections that occurred. And the idea is that we'd be able to do the same thing with humans. And I have a colleague in Sydney in Australia who now has permission to try human trials for exactly this sort of technology. And I'm very excited about that. And that's one of the reasons I've not yet retired. So, few conclusions. Antibiotic production evolved in microbes as a selective advantage. Antibiotic resistance is there because antibiotic production has been present in natural populations of microbes for millennia. But our discovery of antibiotics in the 20th century and their use in hospitals has created lots of new selective islands where resistance can be selected. Horizontal gene transfer allows resistance genes to accumulate in individual cells and often on mobile genetic elements. I hope that our understanding of that process may be an Achilles heel, which will allow us to attack those multiply resistant organisms. It's understanding the evolution and the pressures that has helped us to get to that point. I'll stop there. Thank you.
Fantastic. Thank you, Professor Thomas. We have a couple of questions come through in the chat. If anyone else feels they want to ask a question, there's still plenty of time. One person has asked something very quickly, if they could, they're working on an EPQ topic, they say, related to lecture, and if it would be possible to contact you for further information. Is that possible for those who are interested?
Yes, I'm very happy to answer questions, and Zoom has made it very easy now. If something is too complicated to put in a few sentences, I'm very happy to have a 5-minute Zoom with a pupil. So,
well, fantastic. Well, thank you very much. We have a question from Mr. Ray as well. How close are researchers to the limit of different antibiotics that could be developed for serious bacterial diseases?
We don't know that. First of all, people are working on how you find new antibiotics on two levels. One is still probing natural populations of microbes and coming up with more and more exotic places where you might look. The number of, at conferences, the number of slides I've seen or pictures I've seen of people in scuba diving equipment in some tropical location with the excuse that they're trying to find new microbes that haven't been discovered before. I think it's really just a holiday, but not, I'm joking there. But people are looking for all sorts of places where there might be bacteria that we've not discovered before and that might have been producing new antibiotics. But also that pathway that I showed you, the mupirocin pathway, it's, let me just go back up to there. So you can possibly see that the pathway is made up of these modules that extend the chain and do different things. So you end up with a sort of factory that makes a particular molecule. There are a lot more possibilities than we've discovered so far, in the sense that we think if you can combine those modules in different ways that we've not found already in nature, we will generate new families of molecules that may have new biological activities. So people are also looking at synthetic biology to generate new gene clusters that will make new molecules that have not been seen before. So I would say that we're working on the basis that we are not at all close to the limit of the new molecules that could be discovered.
Fantastic. Thank you very much. We have another question asking from someone in the audience. Do you think it is ever possible to completely get rid of antibiotic resistance?
I think that's very, very unlikely, because even if you, within a herd of pigs or in a hospital, manage to get rid of, or a ward or whatever, get rid of the antibiotic resistance genes, there will still be the reservoirs out in the environment. And so I can see how our probiotic approach, if it works, I mean, if the plasmid can spread fast enough in the gut, and we could clear out the gut as a reservoir from one particular patient so that they are much less likely to get a resistant infection. But the contact with other people will always provide a danger. So I think that you might, well, it will be a constant battle to keep the areas that are sensitive, I mean, that are vulnerable, free of resistance genes. I don't think you'd ever get rid of them from the environment. And it's surprising where things will come from. You know, the water supply, despite in the new QE hospital in Birmingham, very close to the university, one of my colleagues, his group was looking at the resistance. Well, they were sequencing all the Pseudomonas aeruginosa in the burns unit. And I asked them, well, had they looked for plasmids? They hadn't. When they looked for plasmids, they actually found some, like the ones I'd been working on for a while. And one of those plasmids carried resistance to chloride, sodium chloride, which is not very different. I mean, sodium chloride is used for treating water, to purify water. So plasmids can give resistance to water treatment reagents. So combine that with antibiotics, antibiotic resistance, you've got the perfect sort of stealth way of getting back into a hospital. So I think it's a tall order to get rid of resistance genes altogether.
Fantastic. And I suppose there'll always be mutations as well, even if we somehow did manage to get rid of all of them.
Absolutely. Absolutely.
Fantastic. We do have time for a couple more questions if anyone wants to pop any in the Q&A section. Which give you a few seconds to do that if you so wish. Whilst people are giving me a chance to do that, I'd just like to say thank you very much for that talk. It was absolutely fascinating, very much indeed so. Yeah. But it does